be given in a separate, ano siya, in a separate activity, in a form of quiz. Okay, so if walang question, let's now discuss microbial growth. Okay, so next slide please. Para akong nagle-lecture sa conference. Sorry class, I don't know what happened to my computer. Kainis ito. Ito talaga pag nag-change ka ng unit, nagkakaproblema ka. Okay, so this is the outline of the discussion. We'll talk about growth of bacterial cultures, bacterial growth curve and the phases of bacterial growth, and then the direct and indirect measurements of bacterial growth. Okay, next slide. So in our previous module, we have mentioned that the microorganism grow by binary fission. Okay, so that division of or growth of microorganism has requires time, no? And that time required for the cell to grow and divide and to double its number is called the generation time or the doubling time. And it is highly affected by different conditions. Of course, one of the major factor that could affect bacterial growth is the presence of nutrients, availability of nutrients, and of course, the temperature. Okay. Most bacteria have a generation time of 3 hours. Others have 24 hours. May iba pang mga microorganism that takes around more than 10 days for them to double their number. Okay? So, ang example natin, ang E. coli, it can grow to as many as 1 million cells, no? From a single cell in 7 hours and to 1 billion cells in 10 hours and to 21, hindi ko mabilang yung number, 21 zeros in 24 hours. Yan yung E. coli. That's how fast E. coli multiplies. Ayan. So next slide. So this shows the bacterial, the second figure here. Makikita niyo ba yung aking cursor? Hindi kasi hindi ako nag-share ng screen. You can see my cursor. Ayan, sige. Anyway, the second, yung purple na graph is the one showing how microorganisms grow in number, no? Yung yung ginagamit natin is the logarithmic scale instead of arithmetic scales. So the reason for that is bakit tayo gumagamit ng logarithmic scale? It is because bacteria grow rapidly in number. So a logarithmic graph allows us to visualize the complete growth curve. Kasi if we use the pink one, the pink curve, lalagpas sa papel yung ating graph. Okay? Next slide, please. Alright, so ito na yung explanation kanina. Why do we have to plot bacterial growth on a logarithmic scale? Okay. So a arithmetically plotted line does not clearly show population changes in the early stage of growth curve. In another 1 or 2 generations, graph to the same scale would greatly increase the height of the graph and take the line off the page. Okay, so that's why we use the logarithmic scale. Next, please. So this one shows the typical bacterial growth curve. So as you would notice, no, the y-axis shows the log number of bacteria and the x-axis shows the time. Teka klase, nahatching ako. May flu kasi ako kaya medyo hindi maganda yung aking boses. And from time to time, may tumutulo. I'm sorry. Ayan. So a bacterial growth curve has 4 phases. We have the lag phase with an A. Okay, we have the log phase or exponential growth phase, and the stationary phase, and the fourth phase is the death phase or the death or logarithmic decline phase. So each of these phases are explained further in the next slides. Okay, so please. Madami-dami itong lecture sa module 4. It has 3 lectures din. All right. So ito, ayan, thank you for bringing me to this slide. So this shows the more detailed na graph of bacterial growth. So portion A here shows the lag phase wherein there is no net growth, okay? So for example, imagine if you have your bacteria taken from the refrigerator and it was stored there for, let's say, 1 month. So, 'di ba, medyo nangayayat yung bacteria mo doon sa ref kasi medyo na-exhaust na niya yung kanyang nutrients sa media or something. Okay? So, nilabas mo siya ng ref. You want to grow it sa fresh media. Okay? So, ang tawag doon, if it's taken from the refrigerator and you want to culture it again, it's called revival of the bacteria. So, you want to revive your bacteria na naka-rest siya for a while sa refrigerator, you put it in a fresh media. When you say fresh media, madaming nutrients. Okay? So, upon inoculation, it will undergo this A graph. Kumbaga, wala pang growth and then there is no increase in number. By the way, when you say bacterial growth, it refers to the increase in number of bacteria. There is no, it does not refer to the increase in size kasi hindi sila masyadong nag-increase sa size. We are counting their growth or considering their growth in basis of their number. Okay? So with in here in graph A, there's no net growth, and this portion of the graph, yung red sa B, shows an increase in mass. When you say increase in mass, kasi payat sila, nagkaroon ng nutrients, parang meron palang state of nourishment. Hindi pa sila nag-increase in number kasi for them to divide, dapat healthy din sila, no? Maganda din yung health status nila. So, at that stage, ano pa lang yung ating ang bacteria, nag-gain pa lang ng nutrients. So, there is some sort of increase in mass, okay, and volume ng bacteria. Okay, and in graph C, there is this is what you call the log phase or the exponential phase. There is a rapid increase in number. So, dito yung rapid na cell division. Okay? And then, it will approach graph D or the deceleration phase wherein unti-unti marireach na niya yung equilibrium. So, there is still a net growth, but some of the microorganisms are already starting to die. Okay? And yung E na graph natin, that's the stationary phase wherein the net there is no net growth anymore. Balance yung ang cell division and death. Okay? So balance yung nabubuong new cells and yung namamatay na cells. In this portion here, the F, no, which represents the death phase or the the death phase, shows or implies that the nutrients in the media were already exhausted and the microorganisms are already competing with whatever available nutrients yung natira dun sa media. So there will now be rapid decline in number of the microorganism. Ayan, next slide please. So ayan, next tayo. So ito yung mga elaboration nung part nung graph. So the lag phase can take yeah, it varies, no? Depending on the organism. So it can take as fast as 1 hour to several days, depende sa organism. So ito yung tinatawag na acclimatization stage. Wherein your bacteria from refrigerator, for example, acclimatize itself to a new environment, new temperature, and new pH, whatever yung nilagay mo na environment sa kanya, new environment sa kanya. So it will acclimatize there. And then eventually it will start to grow in number. Ayan, so it was mentioned here that an increase in bacterial mass per unit of volume, no? There is an increase in bacterial mass per unit of volume, but no increase in cell count. Yun yung sinasabi ko kanina na nagpapataba pa lang si bacteria, no? Nagsisynthesize sila ng mga enzymes nila and nagme-metabolize, ganun. So that it will be ready for the next phase, which is the log phase or the exponential growth phase. Ayun, next slide, please. Log LAG, the second phase. Next slide, please. Yan na ba yung next? Okay. Hindi ata ito na change. It should be kasi yung kanina is log with an O. This one should be the log. Lag or A. Ito, A. Ayan. LAG. Ayan. Na-change mo? Yung una is LOG. Yung second natin is LAG. Yung second ang LAG. Baliktad ata. Ayan. Yung first is LO. Ay, teka. LA yung first, LO yung second. Ano ba yan? Kahalo-halo. Okay, so log. Aray. Log means logarithmic. Mabilis. Okay? So the cells begin to divide and enter a period of growth or logarithmic increase. So bacterial population increases rapidly in time with an exponential growth in numbers. So yun yung sinabi ko kanina. So take note, cells are most active metabolically. Thus, it is the stage wherein the microorganisms are preferred for industrial purposes. Like for example, kung gusto mo mag-produce ng fermented products, ayun. So dito ka na phase kumukuha ng iyong inoculum. And also, this is the stage wherein microorganisms are withdrawn for microbiological assays like mga drug sensitivity test or antimicrobial activity test. Ganon. And pag tina-target mo yung microorganism or bacteria with certain antibiotic, you usually yung tina-target sila at this phase, no, during their exponential growth phase. Okay, next slide, please. Okay. At this phase, no, the growth rate slows, the number of microbial deaths balances the number of new cells, and the population stabilizes or it reaches its equilibrium. Okay, why is it na, di ba kung tutuusin mo, at stationary phase, medyo nagwi-weaken na yung ibang microorganism, and they start to die na, no? Why is it that you have to target if you need to administer mga antibiotics, bakit dito ka magta-target doon ka sa log phase magta-target, not sa stationary phase? Ano kaya ang reason? Bakit doon ka sa active sila, hindi dito sa portion na medyo namamatay na, mabilis na mamatay? Ano yung reason natin? It is because at this stage, some microorganisms or bacteria are already producing their spores and toxins. Ayan. So pag nag-perform na sila ng spores, it's going to be difficult na to attack, no, with antibiotics. Ayun. So with an exhaustion of nutrients, accumulation of metabolic waste, and harmful changes in pH, the growth rate has slowed to the point where the growth rate equals the death rate. Okay. Next slide, please. So this is now the death phase of the fourth phase in bacterial growth curve. Okay, so the number of deaths eventually exceeds the number of new cells formed. So the reason why madaming namamatay because of lack of nutrients na, and then formation of toxic metabolic byproducts that can cause the death of some microorganisms. And also bacteria tend to autolyze, no? So that's the reason why when we view the when you do turbidity reading of your microorganism, no, in the lab, diba, you used a modified spectrophotometer. So how would you know that your culture is already at its death phase? So diba, at the start you have an increasing curve, or yung reading mo is magsa-start siyang mag-decrease. Okay, and then eventually mag-increase yung iyong absorbance reading. So the indication of that increase in absorbance reading is that some of your microorganism in your culture is already or have already autolyzed. Kaya nagsa-start nang mag-clear yung culture mo. Okay? Next, please. Okay, so we'll now go to the measurement of microbial growth. Microbial growth is measured in cell number or mass, which is often directly proportional to cell number. So we have different methods of measuring microbial growth. We have the direct, and we also have the indirect. Okay. So the population numbers are usually recorded as the number of cells in milliliter. So cells over ml. Yun yung ano natin. Yun yung unit na ginagamit, or cells per gram of sample. All right. Since we cannot measure the population of all of our cultures, we just take a sample, and that sample will now represent the population in the entire culture. Yan yung ginagawa natin, because we, like for example, if you want to measure the microbial load of a pond, for example, shrimp pond, hindi mo pwedeng kunin lahat ng tubig sa shrimp pond and i-view under the microscope or i-count mo lahat ng microorganism doon. You just take a representative sample from that water, no, from pond water, and then you count the bacteria, for example, and that would already represent the number of cells per ml of pond water. Parang ganun yung explanation yan. Next slide, please. Okay, so the first direct method of measurement, or the direct means of measuring microbial growth is the plate counts. So ano yung ano natin sa plate count? So of course you use the Petri plate. No, it's frequently used method of measuring bacterial population, and it gives you the measure of the number of viable cells. Bakit viable cells lang ang ma-measure natin? Because of course, only the viable cells will show the colony doon sa ating Petri dish. The non-viable cells will not form colonies. Hindi sila mag-grow doon, okay? However, plate counting will take around at least 24 hours, no, for you to be able to see the colonies. Okay? So take note that ang single colony that you would see in plate, pwede tayong pumunta sa next slide. Just to show you one of the plates. Ayan. So, yung nakita nyo yung mga bilog-bilog dyan sa plate. Punta tayo sa fourth picture. Ayan. Yan. Makikita nyo, may mga single, ano dyan, di ba? May mga dots dyan. Yeah, tama yung pinupuntahan ng iyong cursor. Magaling pala ito. Si Miss Nufable. Thank you. Ayan. This does not represent single bacterial cell. Okay? This shows a clump of bacteria. Madami yan. Kahit maliit na maliit yan, it does not represent a single cell of a bacterium, no? It represents short segments of a chain. Kung chain yung bacteria natin, like the coccus or staphylococcus, or a clump of bacteria. Madami yan sila. Kahit maliit yung colony na yan. Okay? So, in order to reflect naman the count in the Petri plates, no, during plate counting, we use the unit colony forming units to indicate that the number of colony that formed within the Petri plate. It does not tell us the number of cells in the Petri plate or Petri dish that have grown after 24 hours. Okay, so we usually do serial dilution. No? What is serial dilution? This is used to decrease a bacterial concentration to a concentration or to a required concentration for a test, or to make your sample, to have your sample countable, to have a countable number of cells in your agar plate. Kasi pag, for example, if you test a fishery product, tapos yung fishery product mo is a fermented product. So we know that a fermented product has relatively high number of microorganisms, right? Or bacteria in particular. So you need to dilute your fermented product sample. Okay, so to dilute that, you need to dissolve your sample in, for example, dito sa tube na to, yung nakikita natin sa first tube, yung original na inoculum is, halimbawa, shrimp paste. The shrimp paste is your original inoculum. You put 1 gram of your shrimp paste dito sa 9 ml na water, and then you shake it very well. Okay? And then you take 1 ml, you transfer it to the second tube. So lahat ng tubes na to initially contains 9 ml na media, for example, or broth. All right. So you take 1 ml from your first tube and then you transfer it to your second tube. You shake well your second tube, take another 1 ml, you transfer it to the third tube, and so on until you reach a point na diluted na yung sample. So for every dilution, okay, so for every dilution, you have to have you have to plate your samples. So may plating ka dyan. And you have to look for plates that have 25 to 250 colonies or 30 to 300 colonies. Sila lang yung kino-consider for counting. Okay, in the laboratory class, you will be taught on how to compute for the CFU. Okay, so as I have mentioned earlier, we will not include computations in our exam, no, but I will give you an exercise in a form of a quiz to compute for the CFU. Pero we will not do that this week because of course you will prepare for your exam on Monday. Okay? So the idea of serial dilution is the original inoculum is diluted in a series of dilution tubes. Each succeeding dilution will have only 1/10 the original of the number, 1/10 the number of the microbial cells as the preceding tube. Kaya 1 ml, 1 ml yung kinukuha natin. So may ano yan, kumbaga may dilution factor tayo dyan. Okay, any question? If none, we'll proceed to the next slide. Etong slide na to is sort of ano lang siya, elaboration of the previous slide. So when you do plating, ayan, ito pa rin siya, pareho pa rin nun. When you do plating, you do it in triplicate. You use 3 plates. Ideally, you use 3 plates. Okay, for every tube na galing dito sa taas, you plate them doon, mag-plate ka ng sample mo sa 3 Petri plates. So could you imagine how many plates you're going to use if you did what? 10 dilutions? So you'll be using around 30 plates. Okay? To avoid contamination, it is advisable to start plating sa lowest na dilution kasi mas konti dito yung microorganism mo. But you have to always mix your tube before you take your sample for inoculation. Kasi baka mamaya nagsettle na yung mga bacteria mo doon sa bottom ng tube, di ba? You will not be able to take a representative inoculum para sa iyong plating or sa pagpe-plate out. Okay? So, any question? Ang expected natin na result after incubation of your plates is the one that has the lowest dilution or the one that was taken from the most diluted tube will have the least number of colonies. Ayan. And the colonies should be increasing as we increase the concentration or your concentration. Okay? Okay, this one shows the pour plate method, no? I don't know if this was discussed in your laboratory class, but I think I discussed this one sa amin sa section 2. Okay, so you start with your inoculum placed on the center of your Petri plate or Petri dish, no? And then it's usually 1 ml yung volume ng iyong sample. You prepare your agar, and while it is still hot, no, not too hot para mamatay yung iyong sample, but not too cold para mag-solidify. Kasi diba, we know that Na solidifies at low temperature or at room temperature. So dapat liquid form pa siya. You pour it sa sample mo. Okay? Make sure that your media is sterile. And then you mix it with your sample or your inoculum, and then allow it to solidify and incubate it in an inverted manner. Okay, so the colonies using pour plate method will grow within the nutrient agar as well as on the surface of the agar. Once sinasabing within, nakasiksik siya doon sa media. Okay? Pwedeng mag-grow siya within sa loob ng media kasi nilagay mo siya nung melted pa yung media mo, or pwede din silang mag-grow on the surface of your media. Okay? Iyan yung one of the major differences nila ng spread plate. One of the many differences. Okay, ang disadvantage nito is it might kill heat-sensitive microorganisms, kill or damage. Okay, and it might also distort the distinctive appearance or other structures of colonies, no, na mag-grow doon sa microorganism, especially if that particular colony grow within the nutrient agar. Okay, next slide, please. Okay, the spread plate method. So the spread plate is you have your solid media and then you add your 0.1 ml, 0.1 ml inoculum or sample. You spread it using your hockey stick. Okay, you can improvise this one, and then you allow it to, you incubate it for it to grow. No, after 24 hours, you check your colonies. Then you will see colonies on the surface of the nutrient agar. Ayun. So walang nag-grow within the agar, only on the surface. Okay, so if I'm going to ask you, what are the major, ano yung mga obvious na difference ng dalawa? Okay, first is in terms of volume, no? For per plate, you use 1 ml inoculum, and then the colonies grow within and on the medium. You are in per plate method, you are giving your bacteria a wider area for growth kasi pwede silang mag-grow within the medium. And when you do yun nga, you are providing more areas for microorganisms to grow, and then you can count the colony forming bacteria in a sample, and will also aid you in the identification of aerobic, anaerobic, yung mga facultative aerobes na mga microorganisms. Kasi those that grow doon sa pinakababa ng media na malayo sa source of oxygen, or yon walang masyadong oxygen, alam mo na those are your anaerobic microorganisms. Okay, I am talking about pore plate, guys, ha? Okay, so yung nag-grow naman within, na malapit naman dun sa surface, those are mga facultative anaerobes or facultative aerobes, and yung iba doon mga microaerophiles. Ayan. However, picking of colonies, no, kapag nag-grow sila within the medium is quite challenging kasi you might distort the colonies and the structures of it. Okay, that's for the pore plate. How about for the spread plate? You use 0.1 ml of the inoculum. Again, the growth is on the surface. You are giving the area for growth of microorganism or bacteria is lesser as compared with the pore plate method. However, isolation is easier and more specific kasi kitang-kita mo yung colony na gusto mong kunin. If you want to transfer it to another media. And yun yung mga obvious difference. So guys, you should be able to differentiate a pore plate, no, from the spread plate method. Okay. Next, please. Naghihirap pala mag-lecture kapag sinisipon ka. Okay, next na tayo. Okay. Next method is filtration method. All right. This one is used when the quantity of bacteria is very small. Ano yung purpose natin na filter natin? That is to concentrate your microorganism. Ayan. So, ang filter is being transferred to the petri dish with nutrient, and doon na pinapagrow yung ating microorganism. Okay, saglit lang guys ha. Please give me 20 seconds. Okay, I'm back. Okay, next, that's for filtration. Ayan. Next slide, please. Ayan. So, yan yung makikitang growth, no? This is usually done sa mga water samples. Ang example, what example can you think na ginagamitan natin ng filtration? Anong example natin? Halimbawa, gusto mong i-test yung purified water. Diba? We know already that minimal na lang yung amount ng microorganism na present dyan sa purified water. So you need to do filtration to somehow concentrate yung bacteria na possibly present doon sa sample. Okay, so the colonies formed by this bacteria are distinctive when a differential nutrient medium is used. So after this, no, after mo siyang ipag-grow sa nutrient agar, you if you want to do identification and downstream applications, you need to grow your microorganisms differential nutrient medium. Okay, so the next one is by the determination of the MPN, or most probable number. So this is a statistical estimate technique, no? Nag-estimate ka lang dito kasi you know that the greater the number of bacterial in the sample, the more dilution is needed to reduce the density to countable levels. So, and the MPN is only a statement that there is a 95% chance that the bacterial population falls within a certain range. So, usually ginagamit din yung MPN sa pag-analyze ng water sample. Alright, so let's go to the next slide, please. Ayan, so this slide shows an example of how MPN is done. Okay, so you have to dilute your sample. So you have your undiluted, and then the 1:10 or 10 raised to -1, that's your first dilution. 10 raised to the -2, that's your second dilution. Yan silang dalawa. So the original or the undiluted, the 10 raised to the -1, 10 raised to the -2. Yan yung third tube. Okay, so you inoculate 1 ml into 5 tubes. So you always have to use, if you have 3 tubes sa taas, you'll be using 15 tubes. Okay? So, you have 15 tubes, no? Meron siyang phenol red pH na indicator diyan. So that's why red siya, and then you allow it to incubate, or you incubate it for several hours, and then you will be able to see results. So the yellow color in the tube would represent the positive result. So now you will be counting the number of yellow tubes. So we have here 4 tubes, and then 2 tubes, and then 1 tube. So that will be a combination of 4, 2, 1. There is a table that you use the MPN table to interpret the result. So we have here in the next slide, we look for the 4-2-1 combination of positives. Ayan. So the combination of positives would tell us that there is an MPN of 26 per 100 ml of our water sample. So yan yung kanyang interpretation. Okay. So now let's go to the direct microscopic count. When you say direct, no, you are looking at the bacteria mismo. Okay. So for the first example, you take a measured volume of a bacterial suspension and place it in a microscope slide. And then if you are examining a mobile or motile na bacteria, anong gagawin mo? Kasi 'di ba hindi mo siya makakount kung patakbo-takbo yung mga bacteria. So, ano yung naiisip niyo na strategy that would help you effortlessly count your bacteria?
Fix into place, ma'am.
Yes? Ano yon?
Like, fix ang bacteria.
Wala ako ano?
Fix ang bacteria by adding some, ma'am.
Mga iodine solution. Okay, that could be one. Okay, that could be one. Another is, ilagay mo siya sa ref. Para ma-immobile. Okay, we usually do that if we are counting mga algae or microscopic algae. Ayan, medyo makukulit din sila, so we also do that. Okay, yun yung strategy. So the main advantage of direct microscopic counting is there is no incubation required. Okay, So take note of the name of the slide that we are using. We use the Petroff-Hauser cell counter. All right. So next slide. So ganun pa din siya, no? Um, you use a counter that has grids, no? And then you count the representative sample of your yun sample mo, you count the known volume. And then there is a computation on that, no? Depende sa ginagamit mo na counter. So, I believe bawat counter, they have their formula kasi iba-iba din yung size nila. So, it takes a little practice before mo siya gamay itong direct microscopic counting. Have you done this in your other courses? Hello?
Sa 101, ma'am,
Yes?
I think, oo, ma'am, nag-count kami.
Ano? Sorry Hindi ko pala,
Yes,
ito nga abungol, ako subong, bungol ako guro, kaya ginasipon ako, pero kahinay sa ako na audio. Pero ang akon dira, okay? Klaro ako dira sa inyo?
Yes, ma'am.
Well, good, okay naman na. Sige, hindi ko kabati sa inyo sabat. Okay, thank you sa response. Okay, next slide please. Okay, so these are the indirect methods, no, of estimating or of determining microbial growth. So we have the metabolic activity measurement, the dry weight, and the turbidity. So next slide, please. So for the metabolic activity, usually, no, so for the metabolic activity, this one is very expensive. All right. And as the bacteria multiply kasi, we know that they produce certain products. So that products can be in the form of CO2 or acids. And those products are being measured to determine indirectly the number or the volume of microorganism na present. Okay, ang dry weight naman is this one is applicable for filamentous microorganisms such as molds, no? Plate count will not measure increase in filamentous mass. Kasi kahit isa lang siya, ang dami niyang filamentous mass, no? So hindi mo magagamit yung plate counting. Okay. For turbidity, can you remember your activity in the lab wherein you use the spectrophotometer? So, paano nag-work ang spectrophotometer? You have a light source and then you have a detector or light detector, no? You put in your sample in between of the light source and the detector, and the amount of or the number of microorganism is estimated based on the amount of light that passed through from your light source to your detector. Ganun yung principle on how spectrophotometer works in determining microbial growth. So as bacteria multiply in liquid medium, the medium becomes cloudy with cells. So habang nagka-cloudy siya, the more it becomes cloudy, no? As it increases its cloudiness, ang implication natin is the bacteria is also increasing in number. So habang nagiging mas cloudy siya, mas konti or mas nagsascatter yung light that passes through your tube or your sample. So mas it will now be difficult for the light to reach the detector. So ano yung magiging reading mo kapag ano pa lang, blank ka pa lang? wala ka pang masyadong ang tawag dito, wala pang masyadong laman yung sample, yung tube mo. Okay, so the expected OD or absorbance would be is explained by the change of light will register as percentage transmission of logarithmic expression. So yung nire-read dyan is yung transmission. Okay, so mas maliit yung matatransmit na light kapag konti pa lang yung laman ng microorganism ng sample. Kapag mas madami, ay teka. Oo, 3% transmission nga siya. So ang OD natin or optical density or absorbance reading natin kapag konti pa lang yung ating sample or blank siya is mas mababa. Okay? Mas pataas ngayon ang ating OD or optical density reading kapag mas concentrated na yung sample. Did you get the same results sa inyong experiment? Na yung blank nyo is mas mababa tapos dinededuct nyo siya doon sa inyong readings? Kasi what's the reason why you're using a blank? Kasi yung media nyo pa lang has already an absorbance reading or optical density. So ngayon, it's yung reading ng blank mo plus yung nung media mo plus yung microorganism mo is not the reading of your microorganism alone. Kailangan mong i-deduct yun, doon yung reading ng iyong media. That's why you need to run a blank. Okay? Next slide, please. Ay, bumalik ako. Ayan, so these are just examples, no, pictures of your spread plate and pour plate. All right, so that's for the first lecture. Let's now go to the second one. Try ko kaya mag-share ulit. Ay, ganun pa din siya. Ayaw. Teka. Sige, pa-share na lang ako ulit ng second slide. Second lecture natin. Paano kaya ito, mamaya? I'm sorry. Excuse me. So we are now here sa generation time. So, sige, para akong ulyanin. No, this one is no longer, will not include the computation part. No, but some of the things here might be included in the exam. Okay, only the computation part. Okay, gusto ko naman marinig yung boses nyo kahit medyo bingi ako ngayon. Please, read the dialogue dito. Yung sa first, about Josmin. So 1 becomes 2, generation time. Okay. Sino yung magvo-volunteer? We need 3. Isa yung sa bacteria, isa yung sa boy, isa yung sa girl. Sige, para may break naman. Sige, si Shred. Tapos si K. Ay, si Shred yung bacteria, si K yung girl ba yung isa? Tapos yung boy natin, sino yung available dyan? Pwede naman na girl ulit.
Hindi,
Sino
kita nun,
nakikita
ma'am,
ko
ang
dito?
file.
Oo, si Riza May. Okay ba yung ano mo? Ayan, basahin natin. 1, 2, 3, action!
Hina daw ma'am ang net ni Riza.
Okay, ready na? Dambal ko, galing nakamute ako. Si Shred yung bacteria, si Riza May yung isang bata, tapos yung isa pang bata, si sino yon? Di ko na ano. Sige, si Nancy.
Hindi batian si Shwet?
Di ta ka mo batian?
Hindi ba tian?
Naghambal na ka mo?
Wala
Ay, sinong boy?
Ay, abi ko akong bacteria.
Sadya niya. Shred, ikaw ang bacteria, karon ka pa yung mahambal.
Oo.
Si Nancy
Oo,
na lang
ma'am.
ang isa, tapos ang isa si Riza
Sige.
May. Si Riza May ang naka-blue si Nancy ang naka-green.
Sige, ma'am.
1, 2, 3, action! With Felix, ha?
Spring.
Sino si Riza May?
Tang ano ka no ma'am ng hina daw net niya ma'am.
Sino na lang mapulo? Si Julian na lang. Si Julian.
I love that earthy smell after it rains.
Wow, with Felix.
You're, you're welcome.
Pardon?
Oh, hi. I'm a soil bacterium, and I'm behind that nice earthy smell. I'm a part of a group called the actinomycetes. Actinomycetes. We hang out in the soil, and when it rains, our spores get kicked up into the air and float around, along with their earthy odor. So really, that smell means you're inhaling zillions of tiny bacterial spores. Spring.
Laba-laba, galing mo, Shwet. Ang balo. Okay.
Thank you.
Sige. Julian, please read yung nasa blue na box. Actually, kayo nag-gatukar ako na allergy. Ayan, that's the structure of a jasmine.
So compounds from bacteria, a group of soil-dwelling bacteria called actinomycetes, secrete the compound
Jasmine.
jasmine, which has an sorry, gid kay medyo blur mamina ang connection. So has an earthy aroma when they produce spores. Rain can disturb the compound from the soil. Human noses can detect it at less than 5 parts per trillion, equivalent to a teaspoon in 200 Olympic swimming pools.
Okay. So, di ba kung haba-haba ang init, tapos gulpi nag-uran, parang may iba tayong nasi-smell, and that's jasmine. Okay. So, yan lang yung gustong ipaabot ng slide na iyan. Let's now proceed to generation time or doubling time. Yan yung sinabi natin kanina, no? When a cell divides into two, no, it's the time required for that to take place is called the generation time. So it does not change until nutrients becomes depleted or toxic metabolic products begin to accumulate. So hanggat nasa log phase siya, LOG na phase, yung growth niya, it will, the generation time will always be the same. Okay? So E. coli it has a doubling time of 18 minutes in nutrient-rich laboratory medium, but in an intestinal tract of invertebrates, it is about 12 hours. So the reason for that is kapag enriched laboratory medium naman yung pinaglagyan mo ng E. coli, very optimum for its growth yung lahat ng nutrients na present doon. Plus you put the microorganism in its optimum incubation time, incubation temperature. That's why konti lang or maliit or mabilis ang kanyang doubling time. Okay, next slide. Okay. So the same information is often expressed as growth rate or doubling times per hour. Okay, so this portion, I think you can already just read this one. A growth rate of E. coli growing with a doubling time of, balik tayo dun, sorry. Doubling time of 18 minutes would be 3.3 doublings per hour, would have 3.3 doublings per hour. So kumbaga, sa isang oras, meron siyang, magdo-double siya ng 3 beses. Okay, can you imagine if it started with just 100 for example? Sige, mag-start nga tayo sa 100. So 100 siya, tapos nag-double siya, nag-100 times 100, tapos another 100. Ilan na yon? Okay, let's count that later kasi meron naman talagang formula for this one. Ambisyosa lang ako. Okay. Okay, this is the generation time for some common bacteria under optimal conditions of growth. So, ano yung mabilis lumaki? Mabilis mag-increase in number? Mababa yung generation time or mahaba yung generation time? Ano yon? So, the one with lower or faster generation time. Ayan, tingnan nyo itong tuberculosis. It takes several hours, more than 10 hours for it to double in number. Kaya yung incubation period ng mga ganitong microorganism matagal din. No? Tingnan nyo ito sa tuberculosis. Ayan. So matagal-tagal siya, mga 12 days before it doubles its number. Ayan. So we have a mathematical riddle. Ay nako, may sagot na agad dyan. Boring tong slide na to. A water lily produces a new leaf each day. For each leaf, it has the day before. So starting with a single leaf, it will cover a 1 hectare pond in 30 days. So how long will it take to cover a 2 hectare pond? So ganun siya. Pagka next day, double na agad. Kasi kaya na niya yung 1 hectare. On the 31st day, mapupuno na rin niya yung 2nd hectare. Okay. Okay, so as a cell divides, the population increases exponentially. Now, numerically, this is equal to 2 because 1 cell divides into 2. So, ito yung formula natin. So, the exponent tells us or is the number of times the cell divided or called the generations. Kung ilang beses siya mag-double. Okay? Ang generation niya. So to calculate for the final concentrations of cells, no, you have this formula. Final concentration equals the known number of the cells or the initial number of cells times 2, constant natin yung 2, raised to the number of generations. So we have an example here. The initial number is 5 cells times 2 raised to 9 times. 9 times siya divide. So 9 generations. And that's equivalent to 2,560 cells. Okay. Next is, to calculate for the number of generations, gusto mong malaman yung exponent. What if yung given is the initial number of cell and the final concentration? You don't know the number of generations. Yung exponent ni 2. So this is the formula. Number of generations equals the log number of cells final minus the log number of cells initial over 0.301. So the 0.301 is what? That is the log 10 of 2. So if you are looking for the number of generations, yung 0.301 is also our constant. Okay ba yon? Okay. So yan yung ating example. Sinisipon ako talaga, guys. Kaya natin to hanggang 3 o'clock. Okay. So, to calculate for the generation time, for double yung time natin for a population. May question ba sa slide na yan? Do you have questions? So, yung 0.301, if you're going to ask, saan siya nanggaling? That's the log 10 of 2. Log 10 of 2. Ayan. So, if ang hinahanap mo is the number of generations, constant na rin natin ang 0.301. Okay? Okay na, quiz? So, for the example, calculate the number of generations needed for Salmonella cells to reach a population of 1,720,320 cells. No? So, you start with your
Final population.
You replace the formula here. So final cells is 1.72320. The log number of cells initial is 100, and then this you use the 0.301. You'll be able to get the number of generations. So the answer is 14 generations. So practicean nyo to, guys, ha, for you to be able to answer our assignment. The assignment for this is your quiz na mga problem sets. So ang quiz nyo, answer to the quiz is due Monday after ng, ano ba yung nilagay ko na date? Basta after na ng reading week. Ganon. Tagal-tagal pa yung deadline nito. Okay, so next tayo. Ayan, so meron tayong another formula, minutes per generation. Gusto mong malaman, ilang minutes does it take for it to double its number? So gusto mo yung specific na time. Okay, so to calculate that, you need minutes or generation equals the time elapsed in minute and the number of generations. Kailangan mo lang ng ganun na information. So, calculate for the generation of 100 Salmonella cells growing for 5 hours, produced to 1,720,320 cells. So, yan yung formula. So, the answer is 21 minutes per generation. You will appreciate this equations, no, when you do your activity already. Kasi mas mapapractice mo yung mga formula. Alright? So determine the effect of a newly developed food preservative on the culture of Salmonella. So praktisan nyo ito if you will be able to get the same answer. So ang question is, calculate the generation time and decide whether the preservative inhibited the growth of Salmonella. Kasi suppose 900 Salmonella cells were grown under the same conditions as the previous example, except that the preservative was added to the culture medium. So tingnan mo if after 15 minutes, there were 3.2. Calculate the generation time and decide whether the preservative inhibited. So kung mas mababa yung generation time, saan ba yung example? Ayan, dito, 21 minutes lang per generation. Andito pa lang ako guys sa slide slide 12. Okay? In the slide 21 minutes yung per generation. Yeah, it takes only 21 minutes para mag-double ang number. Sa second, dito, sa slide 13, naging 76 minutes per generation. Meaning, it takes longer time for it to double its number. So, ibig sabihin nun, the preservative was able to inhibit the growth kasi medyo may delaying factor doon sa growth ng microorganism. Do you understand? Nakuha nyo yung point? Maganda din itong mga question na may mga applications. But since we have a very limited interaction, hindi natin masyadong magawa ito. Ayan. So yung practice time na yan yung mga I don't know if given yung answer dito, but meron kayong quiz for you to be able to practice. Okay? So we still have how many minutes? 20 minutes. Ang dami nung slide sa third Madami yung third slide. Teka, tingnan ko muna. Try ko muna guys if kaya kong mag-share ng slide ngayon. Gusto ko talagang i-share yung... Ay, bakit? Ano nangyayari? Portion screen. Ay, nako. Never mind. Sige. Pa-share ako ulit nung nung third. Bago tayo pumunta doon, magchichika-chika muna ako about the vaccine, ha? Okay. So, okay lang ba? Insert ko na yung vaccine. Kasi part siya ng module 2. We have a discussion about vaccine, okay? So, para na rin maging aware tayong lahat. Binigyan din ba kayo ng survey if you want to have or if you want to get vaccinated or not?
Mga matatanda lang ata, ma'am.
Oo, ma'am. Parents lang naman.
Aray, meron?
Ang parents ko lang, ma'am.
Parents lang. So, if you'll be asked, do you want to get vaccinated? Hindi ako nasa-survey. Nagtatanong lang ako, tapos hindi ko siya i-record. I'm just asking you. Gusto niyo magpa-vaccine? Ano yung mga apprehensions? Bakit ayaw niyo magpa-vaccine? Magpa-vaccinate? Ayaw niyo? Gusto niyo? Guys.
Ayaw, ma'am. Ayaw.
Ayaw. Okay. Actually, last month, ayaw ko rin. Pero last week, gusto ko na. Oo. You need to read further, information about vaccines. And you also have to weigh the risk. Not just, wag muna yung risk about vaccination, but risk also if you don't get vaccinated. Ganun yung tingnan natin. Okay, machichika ako. Very quick. Try ko talaga. 5 minutes lang. So, a coronavirus disease 2019 or also called COVID-19 has a number of vaccines available now in the country. Dito sa atin medyo madami na rin. Ilan na yung pumapasok, no? And these vaccines can help us develop immunity to SARS-CoV-2, the virus that causes COVID-19 without getting ill. No, yan yung yun yung purpose ng vaccine. Okay? Vaccines prompt an immune response so that your body remembers how to fight a virus in the future. And some vaccines, use a whole virus to cause your immune system to respond. Take note, yung ibang vaccines gumagamit ng whole virus. Other vaccines use parts of the virus or the genetic material of the virus that provides instructions for making the specific proteins like those in the virus. So yung ibang mga vaccines, gumagamit lang sila ng part of the virus or the genetic material that provides instructions. So yun yung mga mRNA. Okay, inunahan ko na. So many COVID-19 vaccines involve a spike-like structure in the surface of COVID-19 virus. Kasi yung COVID-19, alam natin na meron siyang S protein, no? So the S protein helps the virus get inside your cells and start an infection. Guys, don't worry. I have a slide here and I will give this to you mamaya. I-upload ko din siya. So yung title nito is Vaccine Development Special Topic. Kasi special topic natin to, hindi to kasama sa module. Alright? So, I have included in this PowerPoint yung mga links to the videos about COVID-19. Maganda sana kung makapag-share ako ng slide. I can share with you yung YouTube video about the development of COVID-19 vaccine and how you can compare. Kaya mo bang i-compare or kailangan ba talagang i-compare natin yung COVID-19 vaccines? Kasi 'di ba, you've heard na merong mga 95% efficacy, meron namang 65% efficacy, yung mga ganun. Have you heard about that efficacy ng mga vaccines? Okay, so with these videos will tell you or will help you understand what that efficacy means, no? It does not automatically say that a 95% a vaccine that has 95% efficacy is already better or performs better than a vaccine with much lower efficacy. Yun. Titingnan nyo itong mga, maiiksi lang itong mga videos. Yung first ko is 4 minutes. Yung second is around 11 minutes. Pero maganda, no? It will really tell you kung ano yung, paano ka mag-select nung vaccine. Alright? And paano ka mag-decide later on for yourself if you want to get vaccinated, anong brand yung gusto mo. Ayan. So just a brief background about vaccine development. So it starts with a research, of course, and then magkakaroon sila ng animal testing ng mga candidate vaccines. So yung animal testing ginagawa sa rat or sa rabbit, ganon. And then there will be phases of trials. We have phase 1, phase 2, and phase 3 trial. So for the phase 1 trial, yung phase 1, phase 2, and phase 3, ginagawa na siya sa humans. Phase 1 is done to establish candidate vaccine safety. No, yung safety and ano yung mga side effects, ano yung optimum dose for the vaccine to be effective, ganon. Yung phase 2 naman, it deals mostly sa efficacy of the vaccine, pero mas madami na yung mga, yung population na tinetest nila for the vaccine. Sa clinical trial na yan. And then we have the phase 3 trial wherein yung vaccine is tested for its effectiveness. All right? So during the clinical trials, meron tayong tinatawag na grupo or population or group that receives the vaccine. Yung totoong nagre-receive ng developed na vaccine. And we also have a group that receives a placebo. And have you heard about placebo?
Yes, ma'am.
What do you know about placebo?
Oo, ma'am. Daw may gamay ko nga daw knowledge of placebo and placebo effect.
Angel? Ano to, Angel?
Yes, ma'am.
Ano to, Angel?
Daw may gamay ko nga nabalaan sa placebo effect pala, ma'am.
Okay. The group that receives the placebo is wala sila yasang vaccine nga na-receive.
Yes, ma'am.
No? Ang ilan lang na yan na-receive is ang daw mga filler lang na siya. Kumaga daw,
Daw kunwari lang ang treatment daw muna.
ginaturukan sila but
Pero
wala
wala
sila
ka
gina-inform that kung ano tong in-inject sa ila is not the true vaccine.
Yes, ma'am.
Kumbaga, it's part of the test na may ara dapat nga placebo group. Okay? And later on, dira na nila lantawon ang efficacy no sang vaccine. So they will know kung pila ang nagka-infection nga ara sa placebo group and pila ang nagka-infection nga ara sa vaccinated group. And then that's how they compute the efficacy. So please watch the video ha. Duwa lang ni siya kabilog. Please take time to watch the video. I can also share it sa chat naton dire if you want. But ari lang siya sa sa slide ni i-share ko later. I-share ko sa LMS, hindi ko siya ma-share dire. Okay? So that's for little background about vaccine development. So let's now go to the types of COVID-19 vaccines. So we have the messenger RNA or the mRNA vaccine. So this one uses genetically engineered mRNA to give your cells instructions on how to make a harmless piece of the S protein found on the surface of the COVID-19 virus. After vaccination, your immune system cells begin making the S protein pieces and displaying them on the cell surface. So, amo na siya ang i-identify sa imo body as foreign. And that's magbi-build ka na sang immunity against that S protein. So, an example of mRNA vaccines are the Pfizer, BioNTech, and the Moderna COVID-19 vaccines. Sila ang gagamit sang mRNA. Okay, so next is the vector vaccine. In this type of vaccine, genetic material from the COVID-19 virus, the genetic material is inserted into a different kind of weakened live virus such as an adenovirus. So lain nga virus ang butangan sang genetic material. Okay, the weakened virus will now serve as the vector or the carrier. Okay, to serve as a delivery system. Pag sinabing vector, siya ang delivery system. Parang carrier siya nung vaccine. And when the viral vector gets into your cell, it delivers the genetic material from the COVID-19 virus that gives your cells instruction to make copies of the S protein. Right. So once your cell display the S protein on the surfaces, your immune system responds, no, by creating antibodies. So ganun din, parang the same din sa kabila, but the vector vaccine uses another weakened live virus. Uses a live virus. Medyo scary ito, no? Kasi may live virus na involved. Anyway, if viral vector vaccines can cause you to become infected with the COVID-19 virus or the viral vector virus. Ayan yung statement. Although sinabi ko na scary, pero the viral vector vaccines can't cause you to become infected with the virus or the vector, yung carrier na virus. The genetic material that that's delivered doesn't become part of your DNA. Yun yung ano natin, yun yung part ng study nila na yung genetic material that's being transferred to you during vaccination does not become part of your DNA. So an example of these vaccines na vector vaccines are the Johnson & Johnson COVID-19 vaccine and the eto ngayon yung Johnson & Johnson, meron na sila. The AstraZeneca and the University of Oxford are also working on a vector COVID-19 vaccine. So, tinatry nila itong i-explore. Pero yung available ngayon is the Johnson. Okay, may clinical trial that's going on ngayon dito sa Philippines. Actually, dito sa Iloilo, para sa Janssen na COVID-19 vaccine. And you will be surprised, I will not give the name of the person, meron dito sa atin sa university that's participating in the they are participating in the phase 3, I think, phase 3 trial of Janssen. O, 'di ba? Nakakatuwa. Hindi ako guys. Sinisipon ako guys. That's not me. Okay. And the third type is the protein subunit vaccine. Subunit vaccines include only the part of a virus that best stimulate your immune system. So this type of COVID-19 vaccine contains harmless S protein. So once your immune system recognizes the S proteins, it creates antibodies and defensive white blood cells, no? And if you become infected with the COVID-19 virus, the antibodies will fight the virus. So, an example ng protein subunit vaccine is the Novavax. Ayan. So, again, there are 3 types, no? The messenger mRNA vaccine of Pfizer and Moderna, the vector vaccine of Johnson & Johnson, AstraZeneca, and yung University of Oxford. And the third one is the protein subunit vaccine of Novavax. Ayan. So, hindi ako endorser ng any of these vaccines, ha. Ayan. And at the end, 10 slides lang naman itong vaccine development ko na lecture. Meron ako ditong pinakitang poster. How mRNA COVID-19 vaccines work. So, meron siya ditong explanation. By the way, the material or the presentation in this lecture were taken from CDC, no? Saan na yun? CDC and sa WHO na publication. So, doon ko kinuha ito sila, no? Yung CDC stands for Centers for Disease Control and Prevention. Ayan. So, I will share this to you later. Ayan. So, that's it for vaccine development. Importante kasi as microbiology students that you are also familiar with how vaccines for COVID-19, kasi yan yung meron tayo ngayon na nagpapahirap ng buhay natin lahat, no, are developed and paano sila, how effective they can be, no? And as a person, you can also decide kung ano yung magiging anong vaccine ang gagamitin mo for yourself to protect yourself from the from COVID-19 infection. By the way, what's the main goal of the vaccines in general, yung general na vaccines? It's not about protecting you from not getting the infection. Kasi in reality, hindi natin napipigilan yun na ma-infect ka talaga ng virus. But the idea of the vaccine is for you to be able to combat the infection. Kasi meron kang antibodies. So yun yung ng vaccine. Yan yung concept niya. You will see that sa video later. Sayang lang na hindi ako makapag-share ng screen ko. Okay, hala, meron pa tayong isang slide. Last slide. And it's quite long. It's very, very long. Pero actually, ano ba yung kailangan kong i-discuss dito? Sige, can you just share the third lecture, i-browse natin very quickly. Sorry, 10 minutes ata yung aking vaccine development. Kala ko 5 minutes lang. Miss Nufable. Pa-share po ng ng third slide. Claire, taga-Miag-aw ka, Claire?
Yes, ma'am. Yes, ma'am.
Taga-Miag-aw ka, good man?
Oo, ma'am.
Tumangod ka ni Ellen Claire?
Hindi.
Ha, hindi? Okay. Gusto ko mani-ask si Rika. Taga San Joaquin ka, Rika? Ara pa si Rika?
Taga-haniway na siya, ma'am.
Taga-Haniway siya. Okay. Sige. Okay. Ay, taga-Haniway. Ay, oo, taga-Haniway. Ganun. Sige. Di, kay may kasi na kidato, i-apelyedo. Pero wala ako kabalo pa. Pero feeling ko daw taga-North, manggid siya. Northern Iloilo. Okay. Sige, dasigan na to, na. This lecture will talks about factors affecting microbial growth. Okay, so ito, sige, next na slide. Growth parameters. So you just have to familiarize yourselves, no, about the requirements of bacteria for growth. Ayan, so meron silang mga physical requirements and chemical requirements. So for the physical requirement, temperature, actually for temperature, for pH, and for salt or saltiness, salt level, meron silang mga optimum, no? Especially for temperature and pH, they have this minimum, optimum, and maximum na requirement. So for temperature, at the very low temperature, there will be membrane jelly, no? And the transport of nutrients will be very, very slow. And at low temperature, wala rin halos growth. That's why if you want to keep your microorganism and maintain its number at certain level, you have to keep them or store them at low temperature para hindi sila dumami. Okay? But at high temperature, there will be protein denaturation. Mamamatay yung bacteria mo. Kasi there will be magde-denature yung proteins nila, yung genetic material nila, and then magkakaroon ng thermolysis. Okay, next slide. Sige, okay na siguro yan, next natin. So you have to familiarize yourselves with this one, no? Madaling i-memorize itong mga slide na ito. Yung graph, no? We have the psychrophiles, yung mga close to zero na microorganisms that grow near 0 degrees Celsius, yung mesophiles, yan yung mga that grows at ambient temperature. We have the thermophiles, yung mga sa medyo warm, and the hyperthermophiles, yun yung mga nasa 80 degrees Celsius and up. Okay, next. Ayan, ito yung explanation. So, kaya yung basahin ng mga yan. Okay na yan. Next. Kasi ang dami pala, 61 slides ito. Ayan. May mga tayo, upper temperature limits na for different microorganisms. And this one shows the food spoilage temperatures. So why is it important for us to determine the growth requirement of the microorganism? Because they are responsible in some way in food spoilage or spoilage of our food. And we have to take note the danger zone, kung anong temperature sila mas nag-grow and mas nagko-cause ng spoilage. Okay, next slide. So that's between 18 to 35, 18 to 50 degrees Celsius. Okay. So bacteria also has pH and microbial growth, pH requirement. Ayan, so close to neutral yung mga ideal pH for them, no? Take note, most organisms grow best between 6 to 7.5 na pH. Alright? But some organisms have evolved to grow best at low or high pH. Ayan. So, but they have some sort of mechanism on how to adapt to the varying pH in their environment. Alright? Next. Ayan. So they are categorized as acidophiles, alkaliphiles, and neutrophiles based on their based on the pH to which they grow. Okay? All right. So we have discussed in our previous lecture that microorganisms can also adapt to the varying osmotic pressure. No? Microorganisms obtain their nutrients in solution from the surrounding water. All right, and cell membranes are highly permeable to water. So water can enter or leave the cell depending on the relative osmotic pressure. So bacteria maintain a positive turgor pressure because the osmotic pressure of their cell content is greater than the osmotic pressure of the environment. So hanapin nyo ano yung ibig sabihin ng turgor pressure. So yan yung adaptation ng microorganism kung yan yung adaptation ng microorganism to survive despite the osmotic difference in osmotic pressure with its environment. Ayan. So next, ito yung explanation ng turgor pressure, no? So the concentration of solutes in the external environment increases, that's outside of the cell, and the bacterium pumps potassium ions and or osmoprotectants such as amino acids and proline into the cell and synthesize disaccharides. So kung mataas yung concentration ng solutes outside the cell, kailangan ngayon ng cell na i-increase din yung sort of yung concentration ng solutes niya within para ma-balance niya yung osmotic pressure. Ma-achieve niya yung equilibrium. Okay? So it tries to create a force within the cell that will push the plasma membrane inside the membrane inside against the cell wall. So ayan, these solutes maintain a higher osmotic pressure inside the cell than the outside kapag ganun yung sitwasyon. And eventually, this will cause damage sa essential enzymes and would eventually result or interfere in the metabolism of the microorganism. So that's why ito yung last na note, adding sugar or salt will increase osmotic pressure outside the cell and prevents microorganism or microbial growth. Kasi nga, magbibuild ng pressure inside the cell, si bacteria, for example, that will result to damage in its essential enzymes and interference in its metabolism. Nakuha nyo ba yon? So if we add salt or sugar, halimbawa sa marinating solution natin, pag may bacteria doon na present sa fish sample natin, most likely mamamatay din siya. Okay? Ito yung elaboration ng pinag-usapan natin earlier. Okay, next slide please. This one talks naman about the salt concentration. So, halotolerant, halophile, and extreme halophiles. Excuse us. I'm sorry. Ay, nako. Pinupush ko lang talaga yung yung body ko for today kasi kailangan natin dahil mag-e-exam na. Okay, sorry. Ayan, let's now go to chemical requirements. Ang mga naman, yung content ng slide na to is Yung slide naman na to is complete, no? Very detailed yung pagkakagawa. I just wanna emphasize yung mga important details na kailangan natin. So, sige, magbabrowse tayo. So, carbon is the structural backbone of living matter. Ayan. So, we know that all organic compounds require carbon. Okay? Sige, next. Yan yung classification of organism based on carbon sources. No? Sige, next. So, nitrogen, sulfur, and phosphorus, yan. Needed by microorganisms for the synthesis of cellular materials. Ayan. Andiyan naman yung mga uses nila. So, you just read the slide, no? Sige, next tayo, please. Ayan, nitrogen is a component or it forms our amino acid. Ayan, sa protein natin. Next, microorganisms also require macronutrients and trace elements for growth. Sige, next tayo. Sige, okay. Bakit kasi ang haba-haba ng lecture na to? Ayan, you need to know this, ha? You need to define what an aerobic is. Aerobic bacteria, anaerobic bacteria, facultative anaerobe, and then ano pa? Microaerophile and aerotolerant. Excuse. Yung aerobic, ay balik tayo. Sige, aerobic siya, it grows close to the cup, no? Yung ano siya, yung malapit sa opening kasi diyan ang pinakamataas na oxygen content, yung sa tube A. Anaerobic grows at the bottom of the tube kasi diyan yung area that has very low oxygen or almost zero oxygen, yung sa tube B. Nasa anaerobic na tayo. And then, yung sa letter C, facultative. So, it grows all over the media. Wala siyang, parang hindi siya nag-segregate at certain part of the tube. And then, the fourth one is the microaerophile. Ayan. And the third one is the aerotolerant. Okay. May class pa kayo? May class pa kayo? Sige, very quickly browse natin. Ayan, toxic forms of oxygen, culture media. Ayan, okay lang naman yan. Kayang-kaya nyo yan. I trust you. Sige, chemically defined. Ayan. So, teka. Ayan, take note that complex media are made up of nutrients such as extracts from yeast, meat, or plants, or digest of proteins from this or other sources. Ano yung examples natin? Peptone. Ayan. So, ang nutrient agar and nutrient broth class, nasa ano ako? Nasa slide ako before niyan na isa pa, complex media, ayan. So, take note that our nutrient broth and nutrient agar, the one that we always use in the lab, is actually a type of complex media. Ayan. Parang yung agar natin is just like a broth, except that agar was added to make it solid at room temperature. Okay? Sige, next slide. Ayan yung mga improvised anaerobic jars, no? Pero ngayon madami ng mga anaerobic culture hood or culture cabinet. So there's no problem with that one. Okay, next na tayo. Next. We have selective media, no? These suppress unwanted microbes and encourage desired microbes. Ayan. So, yan yung selective media. It only selects kung ano yung gusto niyang ipag-grow. Yung ayaw niyang ipag-grow. Okay? So, example is the MSA. Next is, ayan, selective media din yung para sa Vibrio. Ayan. Next. Enrichment media. So, this suppresses unwanted microbes and encourage desired microorganisms. So, it is used to encourage the growth of a particular microorganism in a mixed culture. So, for example, meron kang sample na feeling mo konti lang yung bacteria na present dyan and gusto mo ipadamihin pa konti, lagay mo sila sa enrichment media. Okay? Next is, this one shows how to obtain a pure culture. So, 'di ba, you've done in the lab yung yung straking. Okay, nagsi-strake kayo sa lab. And then in reality, at the end of your in the third quadrant ng pag-strake ninyo dito sa second picture, you will be seeing single colonies. As I've mentioned earlier, that colony does not represent a single cell. No, it is composed of several bacteria, a clump of bacteria. All right. So you take that single colony, transfer to a sterile plate, and then you do the spread plate again or the straking until you come up with a single colony again, and then you do it several times until you makita mo a uniform colonies na. So that's the time you check whether you have isolated or you have cultured a pure culture of microorganism. So paulit-ulit lang ito. Yung sa second picture, kukuha ka ng single colony, isi-strake mo na naman siya. And then, pag grow mo 24 hours, after 24 hours, you get another colony, single colony, strake mo again, until such time na makakuha ka ng pure culture. Okay, next slide. This is how you preserve your bacteria. You store it in negative 50 to negative 95 degrees Celsius. But prior to that, you have to add a cryoprotectant agent. So, ang cryoprotectant agents natin can be a DMSO or glycerol. So, the purpose of that is to prevent cellular damage during storage at very low temperature. Okay, so yeah, nag-add usually sa broth nila lagay yung yung bacteria, and then naglalagay ka ng cryoprotectant agent, pwedeng DMSO or glycerol. You mix it, i-mix mo kasi liquid din naman si glycerol, liquid din yung culture media mo. I-mix mo yon, and then maghahalo-halo na si bacteria, si cryoprotectant agent, and your media. So, you mix it and store your sample at negative temperatures. So, ganon. Usually, minsan, meron pa siyang gradual na storage. You put it in refrigerator and then you transfer it to negative 20. Saka mo pa ibaba the next day sa negative 50. May ganon pa na procedure. Okay? That's to minimize the cellular damage. Next, nasa slide 44 na tayo of 61. Okay, itong biochemical test, guys, please read this ha. Kayang-kaya na ito. Ang ganda ng PowerPoint na to that it included everything that you should learn about biochemical test. So why do we do biochem test, no? So each species of bacteria has metabolic or has specific metabolic needs, and that relies on the different enzymes to fuel those unique needs. Okay, so the presence of catalase, gelatinase, oxidase, urease, for example, can be used to identify the species of bacteria, and the biochemical reactions are used in biochemical tests, depending on the bacteria na gusto nating yung suspected bacteria natin or presumptive bacteria. And based on the result natin, later on, sa last na slide, makikita nyo doon yung Burgess Manual, you will be guided na pag ganito yung result ko sa biochem, sa Sa carbohydrate test ko, ito yung result ko sa protein hydrolysis. Ayan, thank you Thank you, Claire. Ayan, you will be able to see na pag ganito yung nakikita mo na result, ito siya. Presumptively, ito yung microorganism na yon. Pag nag-positive ako sa protein hydrolysis, positive ako sa gelatin utilization, ito posible yung microorganism ko. So that's how you identify the unknown. You will be doing several biochemical tests for you to be able to presumptively identify your microorganism. But syempre, nothing beats the tawag dito, yung DNA sequencing, no? Or nucleic acid-based na identification ng microorganism. Syempre kasi ipapasequence mo na yung DNA, so mas specific and mas mas accurate yung ganun na method of identifying the unknown. Okay. Sige. So you just read this one, but pwede tayong pumunta sa slide number 54. Or doon sa saan na yun? Etong ay, gusto kong tingnan yung IMVIC. Saan ba yung IMVIC? Slide ano pa yung IMVIC? Ayan, slide 51 pala si IMVIC. Ayan. So this IMVIC test is a series of 4 tests, no, consisting of indole, methyl red, and Volksproskauer and citrate. Ayan. So IMVC lang siya, 'di ba? Ano yung purpose ng I? You should know that. Actually, wala naman siyang importance. But at least, no, parang trivia siya, but you should know the purpose of that I there in IMVIC test. Bakit may I dyan? Okay, kasi hindi ko rin naman marinig yung answer. Sorry. I is just I was added, no, just to facilitate the pronunciation of the test. IMVIC. Kasi mahirap na siya MVC. Parang mahirap siyang ganun. So, nilagyan siya ng small I to make it IMVIC. Okay? Joke yun. Hindi. Ganun talaga yung niya. Yung acronym niya, may I siya. So, just for the ease of naming the test. Ayan. So, okay na tayo. Siguro, I think kaya nyo na ito. You just read the slides about the biochem test. Kasi dinitalye na lahat dyan. We'll just have another discussion on this, no? If we still have time after the exam. Okay? So, any question? Tanggalin ko kaya yung ko. Tingnan ko yung Ay, kaya pala. Hello?
Hello, ma'am.
Hi, ma'am.
Mas nabatian ko kamo kapag wala ako earphones. Okay, so do you have concerns or problem or anything? So I'll be waiting your I'll just wait for your answers sa assignment na atong answer and critic. No? You complete that and then on Wednesday, ipopost ko ulit sa LMS. Okay? Nabudlayan ka mo mag-access ang document sa Google Docs?
Wala ko pa, ma'am, na-open.
Okay.
Ma-access ang docs, ma'am, pero ang quiz link lang ang may problem, I think.
Sige. If hindi nyo gid siya ma-access, pasadyahan ako tani kung sa Google Doc. Anyway, nasadyahan man ako sa ginubra ko ato ang quiz. So, maybe you just open the file sa LMS and then just answer ang part na, thank you ma'am, ang part na na gin-assign sa inyo. Okay, and then you just i-send ko lang sa ako na i-message nyo lang, i-paste ko lang, copy-paste ko lang na siya sa document. No need to submit a separate file. But if you think na ang na-assign sa imo na question is medyo requires ang mahaba-habang answer, then you may also submit a separate file. Okay? Yung na yun, yung quiz na yun ay parang fun quiz lang sana yun. Oo, madadali lang yung mga questions.
Ma'am, question.
Yes.
Di ba ma'am sa Wednesday, ang deadline ng yung answer sa questions? Same day din ma'am yung deadline for the critic?
Amo na ang hambal ko tani nga as much as possible mag-answer na kamo early para nga ang that deadline na Wednesday is for both na for the answer and the critic.
Oh, okay ma'am.
Oo. Kaya actually, if naghatag na answer, imo nga assign sa mag-answer, kung mag-critic ka na lang, all you have to do is to give feedback, tapos supplement or argue or whatever. But you can't do that, not unless may ara na siya answer na ginhatag.
Oo nga po.
Gani, di ba? So it's a test of cooperation para sa bilog nga class nga mag-submit na sang answer as early as possible. So kung may ara lang gid ya nga may ara nga mga updates pala nga makapag-answer, ko siya. Tingin ko na siya butang ang file man nga makita nyo tanan.
Sa STS naman, ma'am, daw may forum man kami nag-impose lang namon ang amon na answers sa forum and then ang correspond lang or makritik sa amon answers is ma-comment lang siya sa amon nag-impose.
Amo na. Actually, try ko mag-open. Gini nga ba? Ko ba? 17, ako ang ma-post or pwede man siguro no, kamo na lang kaya ang mag-post ng inyo question tapos mag-answer. Dira na mag-critic ang imo nga critic. Kay kung ako ang mag-post, nami akong kamo ang mag-post kay makahatag na kamo dayon answer.
Yes, ma'am.
Tapos bali, this is what you're going to do. When you post your answer, dapat na-copy nyo kag iupod nyo sa pag-post ang question. Kag ang number. Then, halimbawa, if you're assigned in number 10, what? Discuss the blah, blah, blah. So, na-copy mo na and then, butangan mo lang siya answer. Teacher ba ang nag-post ng question?
Yes, ma'am, pero nag-comment lang man kami sa post na nga isa lang.
Okay, the same yung question mo. Tingin ubra ko ba, hindi ako ayawan, gin-assignan, taka mo kada isa sang question.
Yes, ma'am.
Oo. So, mas okay guro nga kamo ang mag-post, no? Sang question nyo, kag dira mag-answer. Dira naman mag-feedback ang inyong critic.
Yes, ma'am. Sa ma-forum.
Oo. Ma-open ako sang forum bala. Ma-open ako sang forum, no? Tapos dira na
Yes,
kamatayan. Okay lang man sa iban? Do you agree with that one? Amo na naton. Amo naton na. Sige. Class, remember, this will serve as your reviewer. So most likely, most of the questions will be taken from that file. So dapat matapos ninyo siya. Okay? So, sige, hindi na lang kita mag-makeup class sa Wednesday. I-consolidate ko na lang siya. Okay?
Okay, ma'am. Thank you, ma'am.
Okay, God bless. Bye. See you.
Thank you, good ma'am.
See you during the exam. Okay?
Bye, ma'am.
Bye,
Thank
ma'am.
you.
Bye-bye, ma'am.
Ay, wala
Bye,
kayo.
ma'am.
Bye.